So welcome to this demonstration video on our lab F1 flow cytometry analysis. During this demonstration video, we're just going to look through some basic aspects of data analysis with flow cytometry data. So what we're considering is the transformation of our linear data into a log hybrid scale. This is more typically known as bi-exponential transformation. And the reason for this is to address this SE spreading error and how subtraction of the background intensity away from the observed values can mean that we can actually get some negative values, which can create issues if we plot them on a traditional log scale. An example of that is shown on our right. So you see a bi-exponential distribution go from negative numbers, so minus 10 to the 3 all the way to through 0, 10 to the 3 up to 10 to the 5, where the most resolve values actually occupy kind of this area between 10 to the 3 and 10 to the 4. Whereas in a log scale, we go from 10 to the 0 to 10 to the 5. Consequently, the values that are smaller here, in essence, get a higher area across this plot. And if we look at the negative values, what happens here is that the negative values are not separated here. So the negative values would appear down here on this graph, on this y-axis. Vice versa, if you look at the x-axis here, there'd be negative values here. And that also means here on the x-axis. Whereas if we look at the log distributions, negative values get transferred to log scale values close to 0. And this means that we have some values that cluster down here, and they may be incorrectly described as clusters or separate cell types, whereas they might represent the absence of any fluorescence in essence. So this means, a bi-exponential distribution can be a way of getting around this issue. So what we can do is explore some R code to look at that further. So let's switch over to RStudio to evaluate this further. So what we're going to look at is how we can generate some data to look at that represent markers, and we can look at their fluorescence and look at how these values change when you plot them using different ways. So if you look at this first distribution, we're just using R norm to generate values with a mean of 1,000 and a standard deviation of a mean of 2,000 and a standard deviation of 1,000. If you look at just a handful of these, so let's say 10, you can see what these values represent. So you can see then there's different values, and each time we run this, we find we get different values. So we're just going to do that for 1,000 values to create some values, and we're going to add on to these 1,000 values the runIf function. We generate some values that correspond to this range here. So if we look at how that is, we're looking at values within a range of 1 to 1,000 there. So if we look at these values again, they're in that range, and each time we do this, they will change. So what we can do is create a CD3 data set representing a hypothetical marker like that with different values. And we can do the same for CD4, except we're going to do CD4 as a function of CD3 with some more runIf values there. So that will mean CD4 and CD3 are associated with each other, but with noise generated by the runIf function. So if we look then at a histogram of the CD3 values, you can see there how they're distributed from some values that are very small, negative even, up to ones that are very high. If we do the same for CD4, again, you see a wider spread, but it's a more leptokurtic distribution, I think. So if we plot them against each other, plotting CD3 on the x-axis and CD4 on the y-axis, you can see that they're associated with each other, and then we have some values that are negative for CD3. If we re-null this, we'll get slightly different distribution shapes, but they'll slightly similar distributions, but they'll have different shapes. So we could add in, some values that are negative here. Yeah, we have more negative values now for CD3 at least. Let's try that for CD4. Yeah, this is a better representation. Maybe a little bit more. Okay, so let's work with this. We have some negative values there for both ones. So you can try some of these questions yourself. To plot a CD3 histogram, CD4 histogram, and then the CD4 versus CD3 cells, all using log transforms. And what we can do is basically borrow our code above, plonk it in here, and just do log CD3 to see what that is shaped like. So you see there, the distribution is quite a different shape. It's now very skewed, with most of the values between 7, 8, 9. If we do the same for CD4, you get a similar effect. So it's very skewed, and you see that some values are down below 3 and some are at 9. If we look at and plotted them against each other, we just insert the logs there. So you saw the shape before is that there was a correlation between them, but there was quite a wide spread of values. See now, we see that this distribution is now markedly different. The original one was here. Now you can see the axes are here going from 0 to 6,000, roughly speaking, on both axes, some negative values. And now you see that most of the values are scattered in the upper right quadrant. Obviously, there's no clear clusters or groups among this linear data. And here we have, very limited clustering again, but they're very compacted. We can look at this further using double log transforms to get a better sense of how this works. Technically speaking, the log by exponential distribution isn't quite like that, but running the function requires a special object. And we'll get to that when looking at the flow cytometry analysis, but it's important to just get a feel for the data that we're looking at here. So if we do the same distributions, we see that they're more skewed. If we do double logs and the same for the CD4 data, and we can do that again for the same distribution to see how retransforming them in different ways, what effect that has. So now you see that they're more compacted. And what we can do is even put them on log base 10. Yeah, so what you see there is that the shape is the same. So what we had before with the scales is slightly differed. And what you see is that they're all compacted in the upright quadrant. And some smaller values are scattered around below there, which maybe are less interesting. So if we're plotting like this, one way of getting around that would be simply to limit the axes according to the log scaling. The log scale we have here. We do that for the y-axis and the x-axis. You see a better resolution there, perhaps. And that captures more of the data. So you can see this kind of demonstrates that there's different ways of plotting the data, and that's something that practicing with your own data would be useful.